Good evening, students. Welcome back to Zoology class. We have been talking about genetics. In that genetics, we have talked about terminology, what we are going to deal in this entire chapter, followed by monohybrid cross. A brief recap of monohybrid cross, and we shall proceed to deviations from these two Mendelian laws, that is law of dominance and law of segregation. Okay, so before going to that, what we have studied, monohybrid cross. Okay, so see here in this, monohybrid cross, the cross which involves parents differing in one pair of contrasting traits is known as monohybrid cross. And we have seen that when a homozygous dominant plant is crossed with homozygous recessive plant, it has resulted in the F1 generation. All the F1 individuals are heterozygous dominant. And when these F1 individuals are allowed to undergo self-fertilization, it resulted in the F2 generation. And in that F2 generation, we have seen that the dominant and recessive individuals are in the ratio 3:1. Okay, so when out of this, Mendel has postulated, based on the inferences that he has drawn from this monohybrid cross, Mendel has postulated two laws, that is first law, that is law of dominance, and second law, that is law of segregation. Okay, so there are two ratios are there for this monohybrid cross, that is phenotypic ratio and genotypic ratio. Phenotypic ratio is 3:1 and the genotypic ratio is 1:2:1. Okay, now here we shall talk about a small concept of probability. What is this probability? Okay, how can we define probability? We can define probability as the possible outcome of a particular, the probability of outcome of a particular event in the total number of possible events is known as probability. Once again, see here, what is probability means? We can define probability as the number of particular outcomes of an event, the number of particular outcomes of an event divided by total number of all possible outcomes. Okay, so what is this probability? I will define. So here we can define probability as the number of particular outcomes of an event divided by total number of possible outcomes of that event. Outcomes of that event is known as probability. Okay, how we will apply this probability to the inferences of monohybrid cross? See, in the monohybrid cross, when Mendel has crossed capital T small t, see, when Mendel has crossed homozygous tall capital T with homozygous recessive, then it has resulted in the gametes. And when these were allowed to undergo fertilization, it has resulted in the F1 generation with capital T small t. So all the F1 individuals are heterozygous dominant. Okay. Now, when this F1 individual is producing the gametes, okay, when this F1 individual is producing the gametes, what is the probability of the gametes containing capital T allele? Okay, A divided by total number of possible gametes, we can say that their probability. Once again, see here, In the F1 generation, we can define this probability as the total number of gametes. How many gametes can be produced by this individual? 2 gametes. Out of these 2 gametes, how many number of gametes has the possibility of having the allele capital T is said to be its probability. Okay, so see here, capital T, small t. If this individual is producing the gametes, now what is the probability of the gametes having an allele capital T? So we can write it as number of particular outcomes of an event, number of particular outcomes of an event divided by total number of possible events. Total number of possible events. Okay. So here, what is the probability when this is participating in gamete formation? So here, it is producing 2 gametes. It is producing capital T allele is 1. Okay. So here we can write capital T, here it is small t. So out of these 2, so here it is capital T. I am talking about allele capital T. See, it is 1 out of 2 even. That is how many total number of possible gametes means 2 gametes are possible. So that is its probability. So the probability of capital T in the total number of events, it is capital T. Capital T, it is 1 by 2. Even small t also, it is 1 by 2. Okay. So the same law, the same probability can be extended even to the F2 generation. So here it is F1 generation. If the same probability is extended to F2 generation, what is the probability of the offsprings having the particular genotype? So the probability of having the particular genotype, capital T is 1 out of 4. Capital T, small t is again 1 by 2. Okay, or 1 by 2, and small t and capital T is again 1 by 2. Small t is again 1 by 4. Okay, 1 by 4, 1 by 4. Okay. So out of this, what is the probability of the phenotype? What are the probability of these offsprings having the dominant phenotype? Okay, out of these 3 are dominant phenotype, and the one is recessive. So how can we write the ratio? Means by adding this 1 by 4 plus 1 by 4 plus 1 by 4. Okay. So by adding this 1 by 4 plus 1 by 4 plus 1 by 4 is having the dominant phenotype. So it is 3 by 4, and the other is recessive. How we got this 3:1? See, when we are applying this probability laws to this, it is saying that the probability of outcome of a particular outcome of an event by total number of possible outcomes of an event. Possible outcome of an event. Outcomes of an event is said to be its probability. Now, what is the probability of the gametes carrying capital T allele is 1 by 2, and small t allele is also 1 by 2. If we are extending that to the F2 generation, it is 1 by 2 plus 1 by 2 plus 1 by 2. That is in case of dominant phenotype. So this adding of all these dominant phenotypes, okay, the sum of all possible outcomes of an event. The sum of all possible outcomes of an event, it is known as additive law. Is known as additive law, or law of sum of probabilities, or we can write it as law of sum of probabilities. sum of probabilities. Okay, hope I am clear. So here, when we are applying the probability to the genetics here, how can we define probability? Means the number of particular outcomes of an event divided by total number of outcomes of that event is said to be the probability. Okay, now with this statement we can find out how many gametes will have capital T allele and how many gametes will have small t. And if you are implicating the same probability rule for the second generation, then how many individuals will have capital T, how many will have capital T, small t, and how many will have small t, small t. That means homozygous tall, heterozygous tall, and homozygous recessive. We can find that, and by adding the sum totaling of all the plausible outcomes of an event, we can tell it as additive law or sum of, or it can be stated as law of sum of probabilities. The next one is, see, based on this monohybrid cross, Mendel has postulated the two laws, law of dominance and law of segregation. Once again, a brief recap of these statements here. Law of dominance. According to law of dominance, the characters are controlled by discrete units called factors, and these factors today we are calling them as genes. And always the factors occurs in pairs. And in a heterozygous state, one is dominant, the one which is expressed is dominant, and the other suppressed is recessive. This is law of dominance. And what is law of segregation? Law of segregation states that when two alleles of a gene present together in a heterozygous state, they do not fuse or blend in any way, but they remain distinct and they segregate during gamete formation such that the gametes remain pure. Okay, that is law of segregation or law of purity of gametes. Okay, now in that, but these laws in the nature, they have seen variations for this law of dominance. One such condition for the deviation from Mendelian law, it is incomplete dominance. See, incomplete dominance, before Mendel have postulated his laws, Carl Correns have studied this incomplete dominance in antirrhinum species and Mirabilis jalapa. Okay, see, incomplete dominance. How can we define this incomplete dominance? Is the failure of the dominant allele to completely mask the effect of recessive allele, result in a phenomenon called incomplete dominance. Okay. The failure of dominant allele to mask the effect of, to completely mask the effect of, to completely masks the effect of recessive allele. Okay, is known as incomplete dominance. because of the failure of unmasking, because of failure of masking of the effect of recessive allele, it has resulted in the F1 individuals has resulted in the expression of an intermediate character. Usually, according to Mendelian laws, all the F1 individuals are heterozygous and they are exhibiting the character of their dominant parent. But here, in case of this Mirabilis jalapa or in case of Andalusian falls, what they have did, because of failure of masking of the recessive character, it resulted in the expression of an intermediate trait. Okay, so this incomplete dominance is seen in case of flower color of antirrhinum species, commonly called as dog flower or snapdragon, followed by Mirabilis jalapa, which is 4 o'clock plant, and followed by coat color in Andalusian falls. Feather colors in Andalusian falls. Okay, so these are the certain examples for this incomplete dominance. Okay, in incomplete dominance, when a cross was taken between homozygous red color flowers red, which are having capital R, crossed with, when they are crossed with homozygous white flowers. These are white. Homozygous white flowers. Okay, it has resulted in the gametes capital R, smaller. Then it has resulted in the gamete capital R and smaller. So the F1 individual is heterozygous, but it has neither represented the character of dominant parent or recessive parent, but it has exhibited a character which is intermediate between these two parents. So when a cross between red and white flowers, it has resulted in the appearance of pink flowers. Okay, these are pink color flowers. Okay, so this phenomenon is incomplete dominance. Now, when these F1 individuals are by selfing these F1 individuals, what happens? See here, capital R smaller crossed with capital R smaller, it has resulted in the gametes capital R smaller, capital R smaller. Okay, now when these gametes were allowed to undergo fertilization, see here, capital R, capital R smaller. Okay, now it has resulted in, see here, capital R, these are homozygous red, and smaller are, these are white. But other plants, they are pink in color. So it is red flower, these are pink flowers, and these are white flowers. Now, when you take this phenotypic ratio, the phenotypic ratio of F2 individuals in case of incomplete dominance, when we are taking the flower color of snapdragon, see the phenotypic ratio is 1:2:1. And the genotypic ratio is also 1:2:1. This 1:2:1 is red flowers is to pink flowers is to white flowers. Okay. And this is homozygous capital R. This is smaller capital R smaller, and this smaller. Okay. According to Mendel's law of dominance, or according to Mendel's monohybrid cross inferences, the phenotypic ratio should be 3:1. But here, in case of incomplete dominance, the phenotypic ratio is 1:2:1, which is a deviation from Mendel's laws. And one more thing, according to Mendelian law of dominance, see here, the F1 individual should resemble the character of their dominant parent. But here, the F1 individual has neither represented the character of a dominant parent or recessive parent, but it has expressed an intermediate trait, which is again a deviation. Okay, so this incomplete dominance is disproving Mendel's law of dominance. Law of dominance cannot be proved in case of flower inheritance of flower color pattern of this snapdragon. Okay, so the phenomenon in which the phenotypic ratio and genotypic ratio are same is incomplete dominance. If you see the inheritance pattern of even the coat color of Andalusian fowls, the same ratios, feather colors when homozygous black in case of Andalusian fowls. In case of Andalusian fowls, when a black fowl is crossed with white fowl, okay, it has resulted in capital P small b, which is neither black or neither white, but it is blue in color. And when this F1 individual, when F1 individuals are allowed to cross, allowed to undergo self-fertilization, again it has resulted in the same 1:1 ratio of 1:2:1 ratio of black is to blue is to white. So here black is to blue is to white, which are in 1:2:1 ratio, which is a deviation from Mendel's law. Okay, Mendel's law of dominance. Once again, see here, what is what is incomplete dominance? The failure of masking of effect of recessive allele by the dominant allele is known as incomplete dominance. Okay, so this in incomplete dominance, the phenotypic ratio is equal to genotypic ratio, that is 1:2:1. But according to law of dominance, according to Mendel's postulates, the F1 generation should represent the character of their dominant parent, but here it is failed. It can be observed in case of Antirrhinum species, Mirabilis jalapa, and in case of Andalusian fowls. The next one, concept of dominance. So before going to this concept of dominance, if Mendel has studied his experiments on this anti-random species, he might have proposed this law of dominance. Why? Because here the law of dominance has been disproved, which will be asked if Mendel has studied his experiments on anti-random species. Then what happens means he might have not proposed this law of dominance, because law of dominance is not obeyed in case of incomplete dominance. But law of segregation is proved. But see here, law of segregation will be proved because when capital R, capital B, small b is there, or when capital R smaller, the gametes will remain pure such that they receive only one of them. So universally accepted law is law of segregation. Hope I am clear. Out of the three laws, that is law of dominance, law of segregation, and law of independent assortment, out of these three laws, universally accepted law is law of segregation of gametes or law of purity of gametes. Always the gametes remain pure and they remain haploid. They have never shown any blending or they have never shown any fusion of characters. One of the deviation for law of dominance, we can say it as incomplete dominance, and one of the deviation for law of independent assortment is presence of linkage groups. Okay, we shall see what are those law of independent assortment and linkage. But do remember now, law of dominance, the deviation for law of dominance is incomplete dominance. Universally accepted law is law of segregation or law of purity of gametes. If Mendel have studied his experiments in anti-random species, he might not have proposed this law of dominance. Hope I am clear with these statements. Okay. Now the concept of dominance. Okay, what is this concept of dominance? Up to now we are talking about what is a dominant allele, the one which is expressed is dominant, which is suppressed is recessive, that's what we know. But now see here, what is this concept of dominance? Say in order to explain the concept of dominance, I have taken an example of a substrate which is converted into product. Okay, see here, say for example, a substrate should be converted into product. The conversion of the substrate into product is taken up with the help of an enzyme. The conversion of substrate into product will be taken up with the help of an enzyme. Okay. Now, it is the duty or the function of the gene to produce this enzyme. Okay. The gene will produce the enzyme and that enzyme will help in conversion of S into P. The substrate should be converted into product. Okay. Now, a gene will have two alternative forms called alleles. The variant type of a character, the visible variant of a character is known as trait. Okay. Now, this gene will have two alternative forms. Allele 1 and it is allele 2. Okay. I am naming this allele 1 as unmodified allele. Unmodified allele, and here I am naming it as modified allele. Unmodified allele and modified allele. Now, what is the function of this particular gene here? What is the gene product? Here the particular gene product is to produce a functional enzyme which is helping in conversion of the complete substrate into product. So I am talking about the functional allele. Okay, in the first case, the allele, first allele, which is unmodified allele, the function of this modified allele is to produce a functional enzyme. Functional enzyme. And this functional enzyme will convert the substrate into product. Now what is the phenotype of the allele 2? What is the phenotype of the allele 2? Okay. So there are three cases. One possibility, 2 and 3. Okay, see, in the first possibility, the allele 2 can also produce a functional enzyme or less functional enzyme. It can produce functional enzyme or less functional enzyme. Less functional enzyme. Okay, functional enzyme or less functional enzyme. And in the second case, the same allele 2 or the modified allele, it may produce a non-functional enzyme. It may produce a non-functional enzyme. And in the third case, the same modified allele, it may produce no enzyme at all. There is no enzyme at all. Okay. So there are three possible cases are there. One is the unmodified allele producing a functional enzyme. That is one. And in cases with the second allele, it may produce in the first case, it may produce a functional enzyme or somewhat little less functional enzyme. In the second case, it may produce an enzyme, but it is not functional. And in the third case, it may not produce any enzyme at all. Then what happens? How the substrate is converted into product? See, if the modified and unmodified alleles are same, then there is no difference. In these two cases, there is no difference. In the first case, there is no difference between modified allele and unmodified allele, because here such cases are more probability in case of this presence of the genes. But if it is producing the second allele and third allele, see, there is two possibilities. If it is producing a non-functional enzyme or no enzyme at all, then the function of the gene dependent only on the unmodified allele. Once again, see here, the function of the gene is to produce an enzyme which helps in conversion of substrate into product. Here it has two alternative forms. One is unmodified allele and modified allele. Unmodified allele producing the functional enzyme which is converting the substrate into a product. Now, a modified allele, there are three chances for this phenotype of this unmodified allele. First one is it may produce the functional allele or less functional enzyme. And in the second case, it may not produce any enzyme, or it may produce enzyme but it is not functional. Then in these two cases, the phenotype will be affected. The phenotype is affected. When the phenotype is affected, so what happens now? In such case, if the modified allele is producing a non-functional enzyme or no enzyme at all, in such case, the original phenotype is dependent upon the unmodified allele. Okay? The original phenotype is dependent upon unmodified allele. So ultimately, the unmodified allele, which is determining the original phenotype, is said to be dominant allele. Hope I am clear. Ultimately, the unmodified allele, which is determining the original phenotype. What is the original phenotype here? Conversion of complete substrate into product is the original phenotype. So the unmodified allele, which is determining the original phenotype is said to be the dominant allele and the other allele is recessive allele. So here I will write the statement. The unmodified allele which is originally determining the phenotype The unmodified allele which is originally determining the phenotype is said to be dominant allele. And the other alleles and the other contrast alleles is recessive allele. Okay, so see here concept of dominance. What is the dominant allele? Okay, every gene has its own function, that is production of its gene product. How can we say that it has produced its gene product? Means by looking at its phenotype. So here we have taken an example of conversion of a substrate into product which is done by the enzyme. If the gene is having two alternative forms, that is called alleles, one allele is unmodified allele and the other is modified allele. An unmodified allele is an allele which is functional allele and it is producing its original phenotype. That is, it is helpful in production of a functional enzyme which can convert the substrate into product. And the modified allele. There are three possible outcomes of a modified allele. They are, in the first one, it may produce an enzyme, functional enzyme or less functional enzyme. Now in this case there is no difference between a modified allele and unmodified allele because both are converting the substrate into product. Whereas in the second case and third case, what are they? It may produce enzyme but it is non-functional and it may produce no enzyme at all. In these two cases, the phenotype of the gene will be affected. In such cases, the phenotype of that gene is dependent only on the functioning of unmodified allele. So ultimately, the unmodified allele, which is determining the original phenotype of a character, is said to be a dominant allele and the other allele is recessive allele. Okay, so this is concept of dominance. Okay, a few points based on and the entire topic of this week, right from genetics introduction to the incomplete dominance and codominance. A few points I want to share. We shall look at those points. In introduction, see here, what is we have forgot to talk about a term that is gene pool. Okay, so see the term gene pool and genome. Now see here, what is the difference between genome and gene pool? The genetic constitution of a single individual is known as its genome. The genetic makeup of an individual is known as genome. Whereas gene pool means the sum total of all the genes of a population in a given area in that particular time is said to be gene pool. Once again, the difference here, genome is particularly about a single individual, whereas gene pool is talking about a complete population. The sum total of all the genes in a given population, in a given area at a particular time is said to be gene pool. Okay. The genetic makeup of an individual is known as genome. Okay, and followed by here, see here, gene pool. How can we define gene pool? The sum total of all the genes. The sum total of all the genes in a population see here it is individual, here it is population. In a population, in a given area, at a particular time. At a particular time is known as gene pool. The sum total of all the individuals of all the genes in a population is said to be genome. And see here I am talking about gene and allele, and see here the place where a gene is located on the chromosome is known as locus. Now, where these alleles are located, when you remember the fundamentals of this cell cycle and cell division, we have clearly mentioned that the two alleles of a gene are present on homologous chromosomes. What are homologous chromosomes? The chromosomes which are similar in their size and structure are known as homologous chromosomes. Okay, so always the two alleles or the two alternative forms of a gene are located on homologous chromosomes. Never the alleles of a single gene they located on non-homologous chromosomes. Remember the statement: alleles of a gene are located on homologous chromosomes. Homologous chromosomes. Okay, so see here. It is one homologous chromosome, a pair of homologous chromosomes, and these are a pair of homologous chromosomes. These are a pair of homologous chromosomes. Now, allele of a gene is responsible for controlling a character called height of a plant, and that height of a plant has two alleles, two traits, two visible variants are there. Those visible variants are known as traits. So those two traits genetically we are calling them as alleles. These two alleles are located on homologous chromosomes. Okay, they are located on, see here, on the homologous chromosome. This is a pair of homologous chromosome, and this is a pair of homologous chromosome. They are located on the sister chromatids of these homologous chromosomes, that is capital T, or small t, they are located. Never a allele will be located on two different chromosomes. Say for example, if capital T is here, one more capital T, this condition does not exist. Okay, is that clear? This is a pair of homologous chromosome. These are a pair of homologous chromosome. So always the alleles of a gene are located on homologous chromosomes, but not on non-homologous chromosomes. And the reason for variations are because of crossing over, and that crossing over will always occur between non-sister chromatids of homologous chromosomes. Always the variations I am talking about. See, what is genetics? Genetics, it is a branch of biology which deals with inheritance and variations. And why these variations will occur, or how these variations will occur? Variations are nothing but recombinations. Recombinations which are takes place in sexually reproducing organisms. Sexually reproducing organisms. Okay. It is takes place during meiosis. See, meiosis which takes place which is helpful in gamete formation. And this recombination, where it is taking place, how it is taking place, because of crossing over. Because of crossing over. And this crossing over will occur in which phase of meiosis? It will occur in meiosis 1. In that meiosis 1, again prophase 1 is there, which is the lengthiest phase. In that prophase 1, there are 5 substages: leptotene, zygotene, pachytene, diplotin, and diakinesis. In those 5 stages, the crossing over will take place in the pachytene substage. In the pachytene stage of prophase 1. In the prophase 1. Okay? And how this crossing over will take place? Crossing over will always take place between non-sister chromatids of homologous chromosomes. Crossing over will always occur between, see here, these are a pair of homologous chromosomes. And one more, these are also a pair of homologous chromosomes. These are also a pair of homologous chromosomes. Okay, and why crossing over occurs? When crossing over occurs means during pachytene substage, when how? If you observe, these two are sister chromatids and these two are sister chromatids. They are sister chromatids of homologous chromosomes. Sister chromatids. Okay, whereas this is non-sister to this and this is non-sister to this. Okay, and these are also sister chromatids and these are also sister chromatids. But this is non-sister chromatid to this and this is non-sister chromatid to this. Okay, always crossing over will occur between non-sister chromatids of homologous chromosomes. Means how? See here. Crossing over will occur between non-sister chromatids of homologous chromosomes. Non-sister chromatids of homologous chromosomes. Okay, it never takes place between chromatids of other chromosomes. Okay, crossing over will occur between non-sister chromatids of non-homologous chromosomes. Always crossing over occurs between non-sister chromatids of homologous chromosomes. It does not take place between the sister or the non-sister chromatids of non-homologous chromosomes. This crossing over does not take place. Okay, so Mendel. So out of these all concepts, Mendel has postulated some of the outcomes from his monohybrid cross. So what are all these outcomes of monohybrid cross? When there is a reciprocal cross, the F2 individual, when there is a reciprocal cross of F1 individuals, it has resulted in the same type of individuals. And the character which is never expressed in F1 character, F1 generation, it has expressed in the F2 generation, that is homozygous recessive individuals. And he also stated that the pairing of the gametes or the fusion of the gametes is purely by chance and it is by random manner. Okay, so these are the outcomes of monohybrid cross. Apart from that, based on the monohybrid cross, Mendel has postulated his first law of dominance and second law of purity of gametes or law of segregation. And in deviation to that, we have seen today, that is incomplete dominance in snapdragon flower color, where phenotypic ratio is equal to genotypic ratio, followed by what we have seen, that is concept of dominance. And finally, in the concept of dominance, what we have studied, the unmodified allele, which is determining the phenotype of a character, is said to phenotype, original phenotype is said to be the dominant allele, and the other allele is recessive allele. So up to here, right from terminology to incomplete dominance and the concept of dominance is included for this week. And tomorrow we shall have paper discussion on the same topic. Thank you.